The first speaker is already here and prepared. I may introduce Victor Norris to you. He's talking about the order of nothing, the bacterial cell cycle, and water. Let's look forward to your talk.
Okay. Well, thank you very much for being here and for some of you foregoing breakfast, so I hope it's going to be worthwhile. Sometimes when I talk about my work with physicists, a physicist will interrupt me and say, well, yes, okay, but what is the problem? Okay, so if you're unfortunate enough to be a physicist, this is the central problem for me in biology. It's the problem of generating coherent behaviours. We heard a lot about coherence. So my talk's going to be in two parts. In the first part, I'm going to discuss, does a cell use its cell cycle, the process with which it replicates its DNA and divides, to generate coherent but incompatible phenotypes? In other words, behaviours. And related to that, does a population of cells have a coherent diversity of phenotypes? And in the second part, I'm going to try and talk about how water might be involved in the cell cycle and in diversity. But for this specialist audience, it's important to realize that I'm not talking to you about my discoveries with water. I'm simply an elderly biologist who tries to make use of your knowledge about water to understand the problem that interests me. Okay, so an obvious question, but why is coherent behavior important? Well, let's suppose it's winter in Bulgaria and you're on the top of its highest mountain, which approaches 3,000 meters. You want to be dressed appropriately. Alternatively, if it's summer and you're running on a beach in the Black Sea, you want to be dressed in a very different way. Both of these people have coherent phenotypes. This wretched individual is completely confused, completely incoherent. They're going to die of hypothermia on the mountain, and they're going to die of heat stroke on the beach. Okay, so that's not a good solution, and natural selection has eliminated these individuals. Okay, so the question then is, how do cells actually manage to do this, to produce this population, working all right, yeah, of coherent phenotypes? Well, it's clear that for differentiating organisms, it involves the cell cycle. This is a Caulobacter chrysanthus. It's attached to a solid surface with its long stalk here, that's the stalk cell, and that grows. And every so often replicates its DNA, shares it out, and divides to give two daughters, one which remains the same, the stalk cell, and the other one, the swarmer cell, swims off. It doesn't actually grow, but it just swims off in search of pastures new. Okay, so that's differentiation. This is another classic case from another model bacterium. This is a Bacillus subtilis in the process of dividing to give a spore. And the rest of the cell, what would be another daughter, is in fact going to die. It's very generous, this is a good mother, to deliver the contents to the spore, which of course won't grow, but which will survive quite a lot of stresses. And I should say that in the rest of my talk, cell means bacterium, and diversity, I'm just going to look at a subset, which is growth rate diversity. And I make no apologies for focusing on bacteria. I know that we have this amazing idea as human beings that we're at the top of the hierarchy. We are the superior organisms on Earth. It's a complete fallacy. It's a self-delusion. The dominant species on the Earth are bacteria. They made our world. They were here billions of years before us. They'll be here billions of years after we are gone. They tell us what to do. They're inside our guts. There are as many bacteria in our guts as there are human cells. They tell you what to eat. In fact, if you want the meaning of life, this is an aside, but I'll give you the meaning of life. If you eat and defecate in different places, you're actually performing the function for which bacteria have designed you. You're transporting them, nourishing them, and deposing them. And all the rest is trivia. And by the standards of our superior species, they're obviously superior because they kill millions of us every year, whereas we really make no dent whatsoever on this superorganism. Okay, so that's just an aside. Can we go on? Oh yeah, so I do realize this is a diverse organism. I'm sorry, I'm talking to you as a mass audience. But what is the cell cycle exactly? Actually, I'm not sure my colleagues who work on it know, but okay, so the cell cycle. This is a bacterium. In red, you've got its chromosome. So to go through the cell cycle, replicates its DNA, which is just starting here. It has to separate and segregate this DNA into the two future daughter cells, and then it has to divide between them. Okay, that's two new cells, and I'm sort of implying here that this is a kind of differentiation. Okay, so which you've just seen with the Caulobacter and the Bacillus. So the question is, how does a cell cycle generate diversity, even in classically non-differentiating bacteria like E. coli? I mean, when I was a boy studying for my PhD, it was generally assumed that in steady state growth, one E. coli produced two effectively identical daughters. Okay, so bacteria are all the same, all growing with the same growth rate in the population. And in a sense, that view continues, despite a whole slew of evidence that it's actually not right. So we're quite good as scientists in maintaining two incoherent viewpoints. Okay, so I'm going to argue that solving the diversity problem involves these wonderful machines called ribosomes. So a ribosome is really, well, I'm simplifying a lot, but it's one of the primary ingredients or primary constituents that makes proteins by translating messenger RNA. Okay. In fact, the old idea of, have a ribosome trundling along a messenger RNA to make a protein isn't quite right, because you can, as in this illustration here, you can have many ribosomes, this is what these big lumps are, all on the same messenger RNA. I mean, as you can see, they're right next to one another. They couldn't be more densely packed. Moreover, of course, the ribosomes themselves have to be made. In this paper here, what you see, this is the same bacterium. So here you see its DNA, its chromosomes in red. And here in yellow, well, there's a marker corresponding to the manufacture of the ribosomes, or the ribosomal constituents. Okay, this is. In eukaryotes, it's well known that the ribosomes are assembled in a structure in the nucleus called a nucleolus. But this is E. coli, and it turns out that, at least in rapid growth, there's a sort of nucleolus, it looks like a nucleolus, where the ribosomes are made too. Okay. So let me give you a few reminders or new information for some of you about this bit of basic biology. The growth rate, in E. coli growing rapidly, largely depends on ribosomes making the mass of the cell. The mass of the cell is mainly protein. a lot of the cell's mass is made up of the ribosomes themselves. In other words, if you took the platonic cell growing in paradise, it would just be a collection of ribosomes making ribosomes. Now, yes, that's heaven for some people. There we are, a strange idea of heaven. Transcription, making messenger RNA, or RNA from DNA. Translation, taking messenger RNA and turning it into protein, and then very often taking proteins, unassembling them into something else, like a ribosome, incidentally. All these dynamic processes can be coupled so that they take place physically together in the same place. And that makes one sort of what we call hyperstructures, which is a big extended structure of many different macromolecules which serves a function. So in the case of ribosomes, they themselves can be made in an extended hyperstructure or nucleolus. And some of these hyperstructures, due to this dynamic coupling between transcription and translation, are associated with a DNA strand. And there's nothing controversial. I mean, it's grossly oversimplified what I'm saying, but there's nothing controversial in what I've said so far. Okay. So the question now becomes, because one question leads to another in this eternal bit of nesting, this Russian doll, could hyperstructures and the cell cycle together create diversity? I mean, I'm really thinking about a cell. This vision of the cell I'm trying to sell now is the cell as a collection of hyperstructures rather than a collection of enzymes, proteins, and genes. Okay, so one thing that seems pretty reasonable to suppose is that different hyperstructures accompany two strands, different RNA strands. Let me just give you a reminder about a really crucial experiment in biology. We have double-stranded DNA with the Franklin strand and the Wilkins strand. This DNA is replicated, and one daughter cell gets the old Franklin strand, and the other daughter cell gets the old Wilkins strand. And the complementary strand is newly synthesized. This is semi-conservative replication. Conservative replication would entail both of the old parental strands going into one daughter cell and both the new strands going into the other cell. And then you've got a whole range of other possibilities. That was a classic experiment of Messelson and Stahl back in the 1950s, which showed that. So replication is semi-conservative. Now, if you imagine that there's a hyperstructure associated with one parental strand, this daughter, one of the daughters, will receive it, but not the other, and vice versa. So that's what I'm trying to show you here. Suppose we've got this big hyperstructure here, this nucleolus, making ribosomes associated with one of the strands. Then one of its daughters will inherit it along with the strand. And reciprocally, for some other hyperstructure, for example, associated with survival as opposed to growth, will go into the other daughter cell. And this daughter cell doesn't start in the same way as its mother, because it's already inherited. It's got a running start. It's already inherited an ongoing hyperstructure. So you could readily imagine that the rich get richer. I mean, that is an obvious observation from our present world. The rich get richer and the poor get poorer. So positive feedback is almost unavoidable. So what I've written here is this is the growth rate of the mother, medium kind of growth rate, and these are the growth rates of the two daughters. One is growing faster and the other slower than the parental cell. So it's a way of getting a sort of symmetry breaking for our physicist friends. Okay. That's assuming that the hyperstructures are segregated with a strand. Of course, you can say, well, this is all nonsense. Is there any evidence for it? Well, there is, and I'm, in a very egoistical way, I'm just going to focus on our evidence. But there's a lot of evidence from much better groups than ours. What we've done is to use a technique called secondary ion mass spectrometry to look at a steady state growing culture of E. coli, at least as close to steady state as we could readily manage. And what we do is we make use of the talent of this machine. Secondary ion mass spectrometry is, I always see it as a kind of chemical microscope. So it gives you, it scans through a cell. It's a totally destructive technique, by the way. And it examines, in the case of the Chemica NanoSims 50, a small volume of the cell, say 50 to 100 nanometers in diameter. And in that small volume, it will tell you how many nitrogen ions you've got, whether they're nitrogen 15 or nitrogen 14. Nitrogen 15 is pretty rare. Nitrogen 14 is the common isotope. So let's suppose now you do this experiment. You grow it in nitrogen 14. At time 0, you transfer it to a medium containing nitrogen 15, and you continue to let it grow in this new medium. Nitrogen is a fundamental constituent of our cells, and it's present in the vast majority of molecules like proteins, nucleic acids. So as the cell grows, it's incorporating the same proportion of nitrogen, but this time, instead of nitrogen 14, it's nitrogen 15. So if you take samples of the cells at different times after the transfer, the ratio of the nitrogen 15 you've got to total nitrogen is a measure of the amount of incorporation that's occurred, and from that you can deduce how fast it's been growing. Okay, so that's what we've done. And if you use a false color here to reflect this ratios, in this population of E. coli, you can see some cells which are, well, really blue, which suggests they haven't incorporated much nitrogen 15 at all. They've hardly grown, whereas there's a sort of a nice virile red. They've been growing very rapidly. And then, because it's a very quantitative technique, about 5 orders of magnitude, it's linear, the incorporation, and the results from the NanoSims, it's counting of the isotopes. You can count how many, you can calculate the growth rate of each cell and see in your population, how many cells you've got which are growing really fast, apparently a 36-minute doubling time. And at the other end, you've got these really pessimistic cells. Think the world is going to end tomorrow. It's all going to be awful. And of course, they're right, it is. And 130, 120-minute doubling times. This is an enormous diversity. And yet, these cells themselves, the population, is doubling every 64 minutes. And if you do the maths and you work out what generation time you get from this lot by working on the incorporation, you get 64 minutes as well. So you sort of square the circle. An optical density measurement of the whole culture gives you 64-minute mass doubling time. This gives you 64 minutes as well. So that's pretty reassuring. But the world's full of coincidences. Okay, so the question, of course, if you've forgotten why I'm saying this, I'm saying, is this related to the DNA strands? How can I prove that to you? Okay, well, we did something a bit strange. I've got to try and remember why I did that. Oh, sorry, let me just go back and point out to you. If you look inside these cells, you see it's not homogeneous. It's not, as I falsely claimed, red cells and blue cells. You can see they're a bit mottled, aren't they? Well, I sort of had the idea that maybe there were regions inside the cell corresponding to these nucleoli, for example. But it's a bit complicated. Maybe there's intercellular information that we can get out of this. So I thought it would be a good idea to take the mode. You remember, you take each cell, and you take the isotope ratio for the entire cell, and then you say, right, what is the mode of that isotope ratio? In other words, which is the value at which 50% of the pixels are below that value and 50% are above it? Okay, so we're just going to get two colors now, above and below. And if everything were perfectly random, they'd be roughly then distributed over the whole cell, 50% above, 50% below. There are quite a lot of pixels within the cell, by the way. On average, are about 600. Okay. So now we can see, this gives us a way then of looking at how asymmetric the population is. If we divide the cell down the middle, where it's going to divide, okay, we can sort of look to see, are there more yellow ones on the left and blue ones on the right? A potentially fast-growing daughter cell will emerge from this half of the cell, and a potentially slow-growing daughter from this half. You can even make a little index out of it by working out, having a measure of how far from random the distribution you find. You can see here, for example, in these cells, look at all the yellow ones in this half, and all the blue ones here. This is a very asymmetric cell. And then you can take each time point, so time 0 after 2-minute labeling, 4 minutes, 8 minutes, etc. And you can work out, for all the bacteria in your sample at that time, what's the asymmetry, the average asymmetry. And it seemed pretty obvious to me. It's obvious, really, that as you label over time, you go towards homogeneity. Obviously, if you grow it for 1,000 generations, everything's going to be uniformly labeled. So the asymmetry should go down with time. And very strangely, that's not what happened. I mean, if I just show that this is the level of asymmetry. And this is the percentage of cells with a given asymmetry. This is time 0 here, various times increasing. And this is the longest time of all we did, 128 minutes. And as you can see, in the 128-minute population, the cells are far more asymmetric than they are at the earlier times. And that really makes no sense at all. Well, at least it didn't make any sense to me and to none of the others. You can't say that the students cheated because it's exactly the wrong finding. But this is why you do experiments. It's to prove that everything is nonsense that you've thought and believed. So there was one strange thing, a wonderful coincidence. Now, I told you that the doubling time of the population was 64 minutes. And as you can see here, these are our labeling times. The only reason it's 64 minutes here is that's a power of 2. We just did it as a power of 2. 2, 4, 8, 16, okay, 32, 64, 128. It was just an easy way to do it. And so when I see 128 minutes, I know, oh, that's 2 generation times. And in the experiment of Phoscillons and Stahl, it turns out they were just labeling the strands and looking at one old strand going into one cell, the other old label strand going into the other cell. After 1 generation, there's no difference. After 2 generations, they get a difference because you get some cells with the old and the new strands, but some cells with just new and new strands. So a difference after 2 generations is sort of a weird echo with Phoscillons and Stahl. so is this increase in diversity really to do with the strand segregation, which is, in a sense, where we came in and which I'd forgotten about? Okay, so I come back to it. The idea then is that, I mean, I've actually forgotten that there was a connection with the model itself and an old paper which we published saying this sort of thing back in 2003. That's a problem with getting old. Anyway, how can I show you this differently? Okay. So I wrote a program to try and investigate this. What I'm investigating is the following. We have a population of cells, well, just take this single cell here, hyperstructure associated with one strand, a different hyperstructure associated with the other strand. This is a kind of autocatalytic process. It's the equivalent of ribosomes making ribosomes associated with a parental strand. And this is the equivalent of the manufacture in this hyperstructure of the other enzymes in the cell, the ones needed for survival. I won't go into the model itself at the moment, but it allows me to examine, for example, several thousand cells growing, going through their cell cycle and dividing. And I've got all of the information sitting in the computer, and I can work out at any one time. Oh, it's like a chemostat. There's a flow through it. So at random, cells are being eliminated from the population, washed out, and new cells are always being born. So I can look at the profile itself, look at the mass doubling times of the cells present, and the number that there are in the population of cells with a particular doubling time. And as you can see, I can get a really broad spread. Okay. Because all of the information is there, I can do this. I can take a particular cell. Okay, now this is, all the history of this cell is present, going right back to Adam and Eve. So, let me take this cell here. This is a very fast-growing cell. Okay, very short doubling time. And I can look back at its ancestors. Okay, and you can see the previous cell generations, there's been an increase in its growth rate over the previous generations. So each cell has grown faster than its mother. And I look here, this is telling me which strand it's inherited. So over the generations, it's been inheriting this strand, this parental strand, with the attached ribosomal hyperstructure. Okay. So we've had positive feedback over the generations due to strand inheritance. Reciprocally, I can take a very slow-growing cell here, and we can see that over the generations, it's been growing more and more slowly. You know, my dad was pretty pessimistic, and I'm really pessimistic. And I suppose my granddad was pretty pessimistic too. So we've all inherited the pessimistic strand encoding the miserable, slow-growing hyperstructure. Okay. So what? Well, this is I'm embarrassed to say this in public for the first time, so you're welcome to shoot me down. But I do wonder whether this means, this is the explanation why DNA replication is semi-conservative. I'm not aware that there's a really good explanation for this fundamental discovery of Mendelssohn's star. I just put it to you, it's a way of generating coherent diversity. Maybe. Okay, well, I don't need to tell the audience this at all, that the structure of water is different in spores and it's different around proteins. Okay, and it affects a considerable portion of the water inside cells. So I'm not going to stray from my area of incompetence and talk about it. But what I do want to know as a biologist is, does this difference in water involve the dynamics of hyperstructures during the cell cycle? I'm not going to say too much about this, but the holy grail for me is to understand the cell cycle in a new way. Many of my colleagues, the vast majority of my colleagues working on the bacterial cell cycle, at least until recent years, are molecular biologists and biochemists. And they see the regulation of the cell cycle in terms of these wonderful proteins and the genes that encode them, very sophisticated proteins. But at the beginning of life, life's origins, cells had to grow and divide as well, and these proteins in their present form weren't there at all. So it is possible that the regulation of the cell cycle, at a fundamental level, really this explanation lies in the world of physical chemistry and physics, which isn't accessible to me and many of my colleagues. But that doesn't mean that it's not the case. So, okay, maybe there's something exciting to be found in the world in which some of you are masters. Okay, well, I've got plenty of time still, or you want me to shut up soon? Sorry? 5 minutes? Okay, it's going to be a long 5 minutes. Okay, well, this is just a little idea, and I don't know whether it's worth very much, but one of the speakers earlier, Dr. Baganu, this week, talked about lipid membranes and phospholipids. And phospholipids, I think he said, they've got this charged little head and the fatty acid chains. And they can be, if it's nice and relaxed, the fatty acid chains are flopping out like this. But if you push them together, the fatty acids become sort of parallel, and the membrane becomes much stiffer and more rigid, okay, which is what you've got here. Push them together. And a few years ago, I was visiting a friend, a computer scientist in the University of Brest, Pascal Ballet, and Pascal has developed this wonderful multi-agent system, and he wanted something to illustrate it. And I said, well, I have collaborators who work with lipid films, Langmuir films, and they compress them, and as they compress them, the constituent phospholipids start to go from an expanded phase into this condensed phase, and they segregate out, so you get sort of little circles of condensed phospholipids amidst the sea of expanded phospholipids, and so on. It's very pretty. So he put that into his program with a bar to compress the lipids, and what to my delight happened, something quite unexpected, was we got a sort of wave of compressed phospholipids moving through his little sea of phospholipids, sort of a travelling wave. And well, I thought, well, maybe it's there's some sort of possible way to having travelling phases. So having a phase shift in a medium which would continue, go on, and maybe not even peter out, but maybe if there's a flow of energy through the system being sustained, you could get endless waves going through it. So oscillations, it's always fun coining a term. So phase oscillations, Phoscillons. Okay. Is it? Well, I don't know whether it's nonsense or not, by the way. So this is just a snapshot of what Pascal's been doing, where we have the green ones are in the expanded state, the red ones are in the condensed state, and these are snapshots over time, just so I can present it easily. And you can see we're going from green to red, and we'll go back to green again as the lipids are removed. Something important here is to, like the bar in as I mentioned with the Langmuir films, to insert the lipids in a defined fixed zone. And that's biologically not unreasonable, because the synthetases which insert lipids into the membranes of E. coli are localized at the site of division, at least for a time. Okay, so maybe it's not nonsense. I don't know whether it's nonsense or not. Bizarrely, and one of these wonderful examples of coincidence, something happened during one of the earlier speakers' talks, because this is just two-dimensional. But of course, you could imagine this happening in one dimension. You can think of a as gyrase is changing the suprahelicity of DNA. You can imagine a change in the density of change in the suprahelical density of DNA traveling down the DNA. So you get a sort of one-dimensional thing. And I thought, well, maybe with water structures, you could get something also propagating in the 3 dimensions. And the closest I've found to anybody, actually, you think about 4 dimensions, but I guess that's only water, so I can't go down that tack. But I looked around to see whether anybody had sort of speculated along these lines, and I see that Dennis Braid did talk about spreading conformational changes in proteins. So I'm not sure it's that original. But ironically, Jerry Pollock, in his talk about muscle contraction, actually mentioned something about myosin. I think it was myosin having a change in its structure moving along the myosin filaments, which echoes very strangely with this. So maybe I'm not original at all. Anyway, could this happen in cells, and could it be based on changes in water structures? But it's not a question that I can answer. It's one for me to put to you in the audience. And the second one, we've heard about sound in this conference and the relationship between sound and water. I'm sorry, I'm taking too long. Here this is a wonderful old experiment by Michio Matsuhashi. And he took a Petri dish, agar, and the 2 halves of the Petri dish were different. On this side you have sort of bacteria which can't grow because of high salt stress. They're close to the concentration of salt which would allow them to grow, but they can't. And on this side of the plate you've got bacteria which are actively growing. And it looked like there was some sort of signal being sent across, which sort of went over about a centimetre, which helped the bacteria on the other side of the plate to grow. And initially they thought it was a chemical signal diffusing, but then they put a plastic septum between the halves, and then they put them in separate plates, and they finally concluded that it was a sonic signal. Wow, okay. And he followed this up by using a sound generator to help these cells to grow. More recently, I saw this paper here where they found a significant change in the growth rate of E. coli using exposure to specific frequencies of sound. So that's an interesting story. Yep. If the sound were influencing the bacteria and some native oscillatory process. But what could be responsible? Well, oh yes, I am going to. Yeah, okay. Well, just remind you that a ribosome goes through a series of conformational changes as it does its job. It's quite complicated, so I won't go into it. But it's a repeated cycle as it's translating. So we have the majority of the cell's material, in some cases, the ribosomes, all going through the same cycle of conformational changes. Remember that they can be packed together in these sort of nucleoli-like structures. Could they become synchronized? Could the movements of ribosomes become synchronized? I know Jan talked yesterday about synchronized movements, the Pelling experiment, the wobbling of the cell coming out of maybe tubulin movements. But of course in bacteria it's the ribosomes which really consume most of the cell's energy. So they're where I would look. Okay, right. I'm pretty close to the end now. So dancing. Okay, dancing. Well, this is if you've ever seen the Sufi dancers, they're absolutely fantastic. I saw this in Egypt. These are wonderful. Okay. If you get them, Sufi dancers densely packed, they're all sort of nodding their heads together. It's just a wonderful spectacle. And this is for our Bulgarian friends. This is a Bulgarian dance where they're dancing together. You get a problem, of course, if you mix them together in the same place, these two sorts of dancers, and you bring in the walls, you start to compress them. I believe when you start to compress them, like starting to remove the water, maybe you get spontaneous segregation. Okay, so, oops. Yeah, so the questions are, after all, we're full of dancing molecules. Do proteins doing the same dance become synchronized? Do proteins doing different dances spontaneously become segregated? Okay, and I've induced one of my friends in physics, insulting the physicists, to try and at a very, very abstract level to sort of investigate this. So this is just a quick 2D model using NetLogo with the proteins in red and blue. Here you are, randomly distributed. Now, they're going to be in two states, and they've got a choice when they bump into their neighbours. Am I in the same state as my neighbours? That's fine. I've got the same value system. Water's only one sort of water, and we stay the same. Or am I in a different state from them? In which case, well, I'm going to move and segregate with, potentially, with people who think the same. Okay, so we go from an initial random distribution to one in which like minds think alike. But an important element here is to have a small group which are already in the same state working in synchrony. Okay, well, so it's an interesting generator of concepts. Okay, so there we are. Are water structures involved in synchronization and segregation? Really fundamental processes in bacteria. And that's my last slide. This is a schematic of a bacterial cell about to divide. It's two different chromosomes with different hyperstructures attached or associated with them, different membranes around them, dancing differently with different water structures playing different roles. And in between them, maybe different lipid, well, actually true, there is a different lipid domain with, as Nigel Dyer has speculated, different water structure affecting the tubulin-like protein which is involved in cell division. And that's a good moment to come to an end, because Nigel's the next speaker. So are water structures central to hyperstructures, the cell cycle, and coherent diversity? It's for you to answer. And there we are. These are the acknowledgements. Well, my friend Camille Ripolla, a theoretical physical chemist, who's helped me with the program and above all with the right equations. A couple of physicists here who've helped with water and the NetLogo program. Pascal Bally, a computer scientist who's developed SIM cells. And of course, the organizers of the conference, the audience, all the technical support and so on. So thank you very much indeed for your patience. I shouldn't be clapping you.
Sorry. Thank you very much, dear Victor, for your presentation.
Yeah.
Are there any short questions? Because
with short answers.
And short answers, because he let us take part in development of his thoughts. This was very interesting.
Oh, thank you. That's
giving
very
us
kind of you.
insights in the thoughts of a scientist in general.
Okay.
Yes. So are there questions? If not, oh, there is one.
Just a comment. I think you're on the right track, without citing specifics, because various people have shown that any conformational change in a protein, or for that matter, I would guess any macromolecule, involves a change in the local structure of water.
Absolutely. And
so if that is really true, as people have demonstrated, then the changes of water structure should be highly relevant for all of these processes. And also, just you're probably aware that Ivan Cameron many years ago studied the changes in water during the cell cycle. And
yes, I do.
He found dramatic changes that occur in a repeatable way in different phases of the cell cycle. So anyway, go to it.
Yeah, thank you very much, Jerry. Yes, you're absolutely right. There are all these wonderful old experiments that one should revisit. And I mean, I know we've been shown the lock and key model for how an enzyme works, but of course, if you look at the work of people like Steve Benkovich and Pratil Agarwal, you see that an enzyme actually goes through a wide range of conformational changes as it performs its function. And these are absolutely fundamental to distorting the bonds, to binding the substrate, to releasing the product, and so on. The many different steps of an enzymatic reaction. And the advantage of what you said about the water, at least as I see it, although I'm having trouble convincing my physicist friends, you know what physicists are like, is that the changes in water structure around the enzyme as it goes through its conformational changes are a kind of a way of averaging, so that it would allow potentially one sort of enzyme to talk to a different enzyme and say, well, we share the same sort of profile of water changes, so let's get together. So you could imagine that as a way of bringing together within the cell different enzymes which collaborate in the same metabolic pathway and maybe help to build the metabolons so loved of Paul Serrer and that crowd. Anyway, thank you very much for that question, Jerry.
Thank you very much. So it's a pleasure and an honor. If there are no further questions.
That's great.
Are there?
Kill the audience.
There is one. Excuse me.
Yeah.
In one slide, you have information about heavy water. Do you have experiment of your model with heavy water or more values for hydrogen bonds?
You're absolutely right. Well, there are plenty of experiments showing effects of heavy water on the cell cycle. So interesting experiments. I mean, what would be very nice to do would be to look at the effects of heavy water on the distribution of phenotypes, for example, growth rate diversity. You know, people say, well, you can add heavy water, it doesn't make that much difference. But do this experiment. Actually, that experiment has to some extent been done because Wagner's group have a nanosims, the environmental microbiologists, and they've added heavy water, and they show considerable diversity in the distribution of heavy water within cells and between cells. But their interests aren't the same as ours. They weren't trying to get a steady state population. They're looking at different species of bacteria. But this would be a very exciting area to explore, and you could hope, you know, that the different hyperstructures within the cell would contain different degrees of heavy water, and then we really, in your world, would be able to relate water structure, perhaps, and hyperstructure dynamics. Sorry, okay.
So, now we are turning to